How do genes cause muscular dystrophy? Imagine your muscles as a well-built house that needs strong beams and sturdy walls to stay intact. Genes are like the blueprints that tell your body how to build those beams and walls. When these blueprints have errors or mutations, the house can become weak and start to fall apart. That is what happens in muscular dystrophy. It is caused by changes in specific genes that are responsible for making proteins essential for muscle health and strength. These genetic mutations lead to defective or missing proteins, which cause muscle fibers to weaken, break down, and eventually die. Over time, this results in progressive muscle weakness and loss of muscle tissue. A key gene involved in many types of muscular dystrophy, especially Duchenne and Becker muscular dystrophies, is called the DMD gene. This gene provides instructions for making a protein called dystrophin. Dystrophin acts like a shock absorber and stabilizer for muscle cell membranes during muscle contractions. When the DMD gene is mutated, the body produces little or no dystrophin, or the dystrophin is faulty. Without enough dystrophin, muscle cells become damaged when they are used repeatedly. This damage causes the cells to weaken and die, leading to the muscle weakness characteristic of these diseases. The DMD gene is located on the X chromosome, which explains why Duchenne and Becker muscular dystrophies mostly affect males. Males have only one X chromosome, so if that chromosome carries the mutation, they will develop the disease. Females have two X chromosomes, so they can be carriers without showing symptoms but can pass the mutation to their children. The severity of the disease depends on the type of mutation. If the mutation completely stops dystrophin production, it causes Duchenne muscular dystrophy, which is more severe. If some dystrophin is still made, it leads to Becker muscular dystrophy, which tends to be milder. Muscular dystrophy can also come from mutations in other genes that affect different parts of muscle cells. For example, facioscapulohumeral muscular dystrophy involves abnormal activation of a gene called DUX4 in muscle cells. Other forms involve genes that encode glycoproteins or enzymes important for muscle function. These mutations can be inherited in different ways, including X-linked, autosomal dominant, or autosomal recessive patterns. Sometimes, large genes like DMD are more prone to spontaneous mutations, meaning they can change even if there is no family history. Genetic testing is a helpful tool to identify these mutations. It can confirm a diagnosis, provide information about how the disease might progress, and help families plan for the future. In summary, genes cause muscular dystrophy when mutations disrupt the production or function of proteins that keep muscle cells stable and healthy. This disruption leads to muscle fiber damage, weakness, and the symptoms we see in these conditions. Understanding these genetic causes is crucial for accurate diagnosis, management, and the development of targeted treatments. If you or your family are affected, always consult healthcare professionals for personalized advice and care.